Bharat. Say what is electrophoresis? Any one of you? Johnson. Siranjeevi. Vimal.
It is a chromatographic technique
Mm.
by which a mixture of charged molecules separated according to the molecular size.
Okay, good. So principal also says the same what he represented. Can you say once again so that others will get the points clearly?
It
Say,
is a chromatographic technique by
yeah.
which a mixture of charged molecules separated according to the
Mm.
molecular size.
Okay, okay, good. Okay, we'll see the principle, what he said. Mm. It is electrophoresis is a chromatographic technique by which a mixture of charged molecules are separated according to its molecular size when placed in an electrical field. So we are going to separate charged molecules according to their molecular size in an electrical field. Okay. So these three parameters we need to monitor. So to conduct electricity, the conducting medium will be of electrolyte. So stationary phase will be what gel, gel or paper, whatever you are using as a stationary phase will be carrying your sample for separation, and the mobile phase decides what ion is going to be separated, whether cation or anion. So mobile phase will be taken as what conducting medium. That's what we have seen here. What is conducting medium? So mobile phase is conducting medium. Stationary phase will be either paper or gel. Paper means like paper chromatography. What Wattman filter paper you have used, right? The same can be used as paper for paper electrophoresis. If we use stationary phase as gel, then it will be called as gel electrophoresis. So mobile phase will be conducting medium, that is of electrolyte. So which decides the pH of that separation. And then electrodes will be of cathode and anode to give the power supply voltage for the separation, and it is represented with the movement of particles are represented with V equal to m into E. V represents velocity, m represents mobility, and E represents field strength. So this is all we have discussed, the different characteristics of electrophoresis separation. In the last class itself we have discussed. This is the scheme representation of electrophoresis technique. Now let's move on to the second part, that is equipment for paper and gel electrophoresis. So there are two categories. According to your syllabus, you have to learn what is paper electrophoresis and what is gel electrophoresis. Since these two techniques, many techniques are there under electrophoresis based on the principle of separation. According to your syllabus and according to the clinical applications, paper and gel electrophoresis are most important. So you have to learn concepts behind paper and gel electrophoresis. Okay. Where in paper electrophoresis, stationary phase will be paper. In gel electrophoresis, we'll be using as polymeric gel as stationary phase. So based on usage of stationary phase, it is divided into two categories, paper and gel electrophoresis. Now let's see the equipment for paper electrophoresis. This is of two kinds available. Horizontal electrophoresis, paper electrophoresis, and vertical paper electrophoresis. Horizontal means like radial chromatography. In horizontal position we run, no. Likewise, horizontal, it is horizontal paper electrophoresis. So paper is kept as in the flat surface, it is kept, and the sample is spotted in one direction, and when the electricity is passed and the paper is immersed in the conducting medium, you can see that this blue color thing is conducting medium, mobile phase. So the paper will be equilibrated or immersed in the mobile phase, where it is spotted on one side with the samples. Here you can find the markings of samples. And this will be when the electrical current voltage is applied here, it will be started moving and separated according to its molecular size. Okay, this is about horizontal paper electrophoresis. Vertical means the sample will be here, the conducting medium, the spotted paper with sample is immersed in the conducting medium, and it will be start moving like ascending technique. It will be moving up for the separation when voltage is applied. So there are two kinds of paper electrophoresis is available, horizontal and vertical. Okay. So here this cathode and anode will be connected to the conducting medium where it conducts the electricity for separation. Okay, it is a simple concept only. And here we can see the parts and with the description of paper electrophoresis. Here filter paper, stationary phase used is Wattman filter paper or cellulose acetate filter paper or chromatography paper. So any three kind of papers can be used. Mostly Wattman filter paper is preferred. So according to the sample separation, nature of sample, we could prefer any type of filter paper, either Wattman filter paper or cellulose acetate or chromatographic paper. Mostly used paper is Wattman filter paper. What you have used for ascending and circular paper chromatography. In normal paper chromatography, we have not applied any electrical current for separation, but in electrochromatography, we are applying electrical current for separation. So what filter paper is used? Wattman filter paper. Coming to the apparatus of paper electrophoresis, it contains power pack, electrophoretic cell that contains electrodes, buffer reservoirs. That's what you have seen here. Power pack. And the electrophoretic cell, which contains what electrodes that is attached, and the conducting medium, mobile phase reservoir, then the stationary phase bed, where the paper is kept with the sample. Okay, buffer reservoir, support for paper, transparent insulating cover, here, transparent insulating cover over the stationary phase. So these are all the parts of paper electrophoresis. Next, coming to sample application, sample may be applied as a spot with measured. You can use a micropipette for a measured amount or volume of sample spot that to be kept over the paper. Okay, what is the spot distance is about 0.5 centimeter in diameter. 0.5 centimeter in diameter. Around this diameter, spot is applied over the paper. Okay, or as a uniform streak. It is applied over the paper. This is about sample application. Then the electrophoretic run. The current is switched on after the sample has been applied to the paper and the paper has been equilibrated with the buffer. So once the spot is applied over the paper, then the paper is equilibrated with the buffer solution, and then power supply is applied for separation. Okay. Then deduction, physical properties, like what are the properties for the separated molecules we focus means physical properties like fluorescence, UV absorption, ultraviolet absorption, fluorescence, or radioactivity or exploited for deduction. So what are the physical properties we will deduct for the separated molecule? Fluorescence, UV light absorption, and then radioactivity can be deducted, okay, by paper electrophoresis. So for this purpose, if the sample is having this particular physical characteristic, then we can analyze or separate by using paper electrophoresis. What are the applications of paper electrophoresis means? It is applicable for serum analysis for diagnostic purpose. All are listening, right? Can observe the PPT.
Yes, ma'am.
Oh, okay. Thank you. Okay. Applications are serum analysis. It is applicable for serum analysis. Hope you know how to make serum from the blood. For diagnostic purpose is carried out by paper electrophoresis. Okay, serum analysis for diagnostic purpose. Any disease diagnosis is carried out by paper electrophoresis. So it is having more clinical application, paper electrophoresis technique. That is why you are learning from this syllabus. Okay, muscle proteins, then it is applicable for separation of muscle protein, that is myosin, egg protein, albumin, milk protein, casein, snake and insect venoms have been satisfactorily analyzed using paper electrophoresis. So these are the applications of paper electrophoresis, but it has the disadvantage that it is very time taking. Time taking process, around 14 to 16 hours it takes for the complete separation. Okay, how many hours? It is 14 to 16 hours it takes for complete separation, where normal paper chromatography we can separate within maximum of 20 minutes, we can complete in minutes the separation occurs, where for paper electrophoresis around hours it takes for separation. So it is time consuming process, that is the disadvantage, but it has advantage of separating many proteins, and then even serum analysis, the constituents present in serum also can be separated according to its charged, mainly charged molecules present in the sample can be separated. Proteins can exist in both positive and negatively charged molecules, so that since proteins are sweeter ion molecules, it will be having amphiprotic in nature, that is two it can have both positive and negatively charged molecule. So when you increase the pH, it will act as anion. If you decrease the pH, the molecule will act as cation. So according to the pH, according to the isoelectric point PI value of individual protein, we can decide whether acidic pH is required or basic pH is required for the separation. Okay. So here it decides, the conducting medium decides the pH. Electrolyte, if it is you're making electrolyte with basic buffer, then the separation occurs towards anode. Anions will be moving towards anode. If you are making acidic buffer, then the sample should be moving towards cathode. So the sample should be applied towards the cathode, or opposite direction you need to keep the paper, so that it will be moving towards cathode. So you will be knowing that what is the pH of conducting medium, so that you can decide the separation, at what direction it has to move, either towards anode or towards cathode. So likewise you need to apply the sample, and you have to keep the paper in the medium. Okay, in the medium of separation. So this is about paper electrophoresis. We are using stationary phases, Wattman filter paper or cellulose acetate filter paper or chromatography paper, and apparatus consists of the parts what you have seen already: power pack, electrophoretic cell, electrodes, buffer reservoirs, buffers, support for paper. To hold the paper we need a support that you can find here. These are the supports for paper. Then a transparent insulating cover. Transparent insulating cover, why we need to use means to observe the colored bands what it is separated, so that transparent insulating cover over the paper is kept. Then a sample application around 0.5 centimeter in diameter we need to apply, or as a uniform streak you can apply. Taking a uniform volume of spot you can apply for all the spots of sample. Then electrophoretic run is started when the sample is spotted over the paper, and the paper is getting equilibrated with the buffer. Once it is equilibrated with the buffer, then electrophoretic run starts. Then the sample is getting separated towards particular electrode. Okay, according to molecular size. If the molecule, if you are deciding the pH buffer as either acidic or basic, then the molecule also carries that particular charge. The molecule will be either positively charged or negatively charged, and the mixture will be having all negatively charged molecules or all positively charged molecules, and they will be getting separated according to their velocity. That's what we have seen. Velocity equals to mobility of ionic particles into applied strength here. Velocity, and this velocity depends on the molecular size. Okay, if the heavy molecule is having is heavy, then it will be moving very slow. If it is light, then it will move very fast. So according to the molecular size, it will be getting separated towards either anode or towards either cathode. It will be separated into individual spots. So from deduction, how we will detect either fluorescence detection or ultraviolet absorption detection or radioactivity. Through this three physical characteristics detection, the molecule molecule can be detected. Separated molecule can be detected. And what molecule we are expecting that can be detected through this physical detection methods. Okay. And then applications, different particles, biomolecules present in serum can be separated for diagnostic purpose. Any disease diagnosis, it can be separated by using paper electrophoresis. And we can separate many body proteins, myosin, albumin, and milk protein, casein, and venom, snake and insect venoms, all can be separated and analyzed by using paper electrophoresis. Disadvantages, it is time consuming process. Around it takes 14 to 16 hours to complete the separation. Okay, this is about paper electrophoresis. Now let's move on to gel electrophoresis equipment. For gel electrophoresis, it is also having two kinds, like horizontal and vertical gel electrophoresis. Horizontal gel electrophoresis only I have focused here in the principle, right? The same. Okay, electrolyte solution is taken. It is horizontal gel slab, and conducting medium of electrolyte is placed here, and conducting medium will be carried through wick to the slab. And then here you can find the sample wells in gel where the sample is injected. So we need to make, you should have made in microbiology, after making agar plates, you will be making like wells, no, small wells to keep your samples. Likewise, here also small wells are made over the gel slab to keep the sample, to inject the sample. Okay, so uniform or increased concentration of sample or whatever you need to apply, here you can apply in the well. After then the conducting medium, after applying electricity, it will, the sample will be getting separated over the gel. Okay, and gel electrophoresis, comparing paper electrophoresis, is commonly used for clinical sample separation. Okay, but you need to know concepts behind both paper and gel electrophoresis. You need to make a gel, and it is a horizontal gel electrophoresis. It is vertical gel electrophoresis. In vertical gel electrophoresis, what are the parts you can observe means, these are loading wells. In the top portion, it is a vertical gel plate. Okay, here you can find the wells for injecting the sample. This is the micro pipette through which you are injecting, taking the sample, you are injecting the sample into the wells. Okay, and the gel is made with STS gel electrophoresis. STS page cell electrophoresis is widely used. STS represents sodium dodecyl sulfate. Okay, STS is sodium dodecyl sulfate polyacrylamide gel. Okay, so it is called as, when STS is used, STS page gel electrophoresis, it is known as, it is widely used for protein separation. Okay, clinical sample separation. Which gel electrophoresis is widely used? STS page. Page, spelling of page only, PAGE. Page represents polyacrylamide gel electrophoresis. Okay, STS combined with page gel electrophoresis is widely used. What is SDS? Sodium dodecyl sulfate. Sodium dodecyl sulfate with page. PAGE. Polyacrylamide gel electrophoresis. So since you are using gel, that is polyacrylamide gel combined with STS is used for separation. And here cathode and anode you can find for applying the current. Towards anode only the sample is getting separated, since the you will be using basic buffer. So conducting medium will be of basic in nature, so that only your sample will be in nature of anions, and that will be moving towards anode once the electrical current is applied. And here two gels you can find. One is stacking gel, another one is resolving gel. What is stacking gel? Stacking gel means it will be arranging all the proteins to move in uniform way. Okay, stacking gel will make all the proteins or all the samples to move in a uniform way. If we since stacking gel will be having low pH, what it will be having? Low pH, that means acidic pH, around acidic pH, so that at cathode acidic pH means the cations will be there at the cathode. So in acidic medium, the samples will be arranged to move for the separation. So we need to make the gel bed with two types of gel. One is stacking gel, another one is resolving gel. So stacking gel pH will be low pH. What difference these two gels will be having means pH only. It differs with pH. And stacking gel will be arranging the molecules to move in a uniform way. Okay. For that, it is first add and prepared where the wells for applying sample is made. Okay. And once the electrical current is passed, all the protein samples are getting ready uniformly to move. Okay. And then resolving gel, what is resolving gel? Then it will be having highest pH, means basic pH, so that the molecules will become anions and it will be moving towards anode at that pH. For example, it will be having around 9 pH, it will be having around 6 pH, you think. Stacking gel will be of 6, pH will be of 6, where resolving gel pH will be of around 9. So it is basic pH. It makes your sample to become anion, and these anions are getting separated according to the molecular size over the gel plate. Okay. So like this, it is getting separated, and this is observed by either UV light, UV cabinet. You can see the UV absorption or fluorescence or radioactivity, whichever you need to observe for that separated protein, that you can detect and find out the separated molecule. Okay. Even you can go for quantification also. So these are quantification methods only. Even you can recover the separated sample from the gel and you can go for deduction. So this is about gel electrophoresis and vertical gel electrophoresis. And it is the gel is made with glass plate only, transparent glass plates. Since if it is made with glass plates only, we can observe the separated spots. Okay. So this is what gel electrophoresis. So what are the parts? It has electrodes, cathodes and anodes to apply voltage, then loading wells at the top, and here at the cathode junction you will be spotting the samples by means of making a well or a number of wells in the gel. Okay. Gel should be already prepared and it is dried, and then wells also are made during the preparation itself. Then the sample is applied into the well. After then, stacking gel makes the all the samples to run uniformly. And then resolving gel resolves the sample according to its molecular size towards the anode. Okay. And it is made with glass plates for transparent purpose. And then what gel is commonly used? STS page gel. STS represents sodium dodecyl sulfate. Page represents polyacrylamide gel electrophoresis. Okay. So STS page is widely used technique in gel electrophoresis. Okay. Now you can see the flow chart of the process gel electrophoresis. What I have explained, the same is given in flow chart. So first we need to prepare agarose gel. In place of agarose, what you can use other gels. What is commonly used? Gel STS page. For sensitive separation, STS page is widely used. Okay. Agarose is commonly used when you are not having STS page, then general gel will be agarose gel for the separation. Okay. And it should be melt, cool, add ethidium bromide and mix thoroughly. So prepare agarose gel in this manner and then pour into casting tray there with a comb and allow it to solidify. Add running buffer, load samples and marker. Marker will be of your standard samples of your samples what you need to separate and find out. That is standard, will be compared with standard marker. Then run gel at constant voltage. Voltage should not be altered throughout the separation. If you fix some voltage, that will be constant. Okay. Until band separation occurs. Then view DNA on UV light box, or and you can find the results. Okay, you can view the separated spots, either DNA or proteins. Other proteins all can be separated. Macromolecules can be separated and can be detected by using UV or fluorescence or radioactivity detection methods. Okay. Now let's move on to various types of electrophoresis. Here you have, according to syllabus, you have learned only paper and gel electrophoresis, equipment for paper and gel electrophoresis as per the syllabus. So with the content, you can explain what manner you have understood. Even you can take more examples on paper and gel electrophoresis through net also. Many articles have been published in this electrophoretic techniques. Okay. So you can collect different samples, what different samples are separated, and make an assignment also on that. Okay, applications of gel electrophoresis on different samples. Collect different samples like HPLC. You have made an assignment. No, likewise you collect. Material on different articles that is meant for gel electrophoresis, different proteins, different kind of proteins, how it is separated, observe, and what are the experimental parameters they have fixed for separation, that also you note it down and write an assignment. Now I am not assigning, since I have already given an assignment. So whoever is interested, make it. Once you finish it, then tell me, that time I will assign the assignment. Okay, whoever, even tomorrow also you can make it and submit. You can just refer article, just write a written material, then type the same and then send it to me. If all 30 members are sending, it will be good. Or if any 5 persons are sending, then I will consider on applications of gel electrophoresis, pharmaceutical applications of gel and paper electrophoresis. Make a tabular column of what are the contents you require, then stationary phase what gel is used, and what mobile phase it is applicable, if any voltage range, what it is, what is the deduction parameter. This all you make it as a tabular column for 10 samples at least. For gel 10 samples, and for paper 10 samples. Collect from the articles. Articles, hope you know, articles published in Google Scholar or indexed articles. Okay, from that you can collect and write the material and then submit. Okay, and coming to types of electrophoresis, it is of two categories: zone electrophoresis and moving boundary electrophoresis. Zone electrophoresis and listening, right, all of you? Okay. Zone electrophoresis and moving boundary electrophoresis. Zone electrophoresis comprises of 4 techniques: paper, gel, thin layer electrophoresis, cellulose acetate electrophoresis. Here we have discussed these 2 techniques, paper and gel electrophoresis, which are commonly used for electrochromatographic techniques, where other techniques are thin layer, cellulose acetate electrophoresis. Then moving boundary electrophoresis are capillary, isotachophoresis, isoelectric focusing, immunoelectrophoresis. Capillary, where we will be using capillary for the separation. Isotachophoresis is all depending on the isoelectric point of samples. Isotachophoresis movement will be migration will be same for 2 molecules. Then isoelectric focusing, same voltage will be same for 2 molecules. Then immunoelectrophoresis, immunoantigen antibody separation. This all will be carried out with moving boundary electrophoresis technique. Capillary electrophoresis, isotachophoresis, isoelectric focusing, immunoelectrophoresis. These are coming under moving boundary electrophoresis. So electrophoresis is of zone and moving boundary electrophoresis. Okay. Then coming to the third part, applications. So from the explanation itself, you should have understood it is applicable for many kind of biomolecule separation. Starting with DNA sequencing, it is applicable for DNA sequencing, medical research, protein research or purification, agricultural testing. Here I have focused different fields of separation. So you can take means each field, and you can find some examples through articles. Okay. and you can give it as a tabular column for each category. What are all the components are analyzed, so that you will get clear idea what kind of samples we can separate by electrophoresis technique. Okay, that's what I'm asking you to make assignments. So agricultural testing, it is employed in biochemical and clinical fields in the study of protein mixtures such as blood serum, hemoglobins, and in the study of antigen antibody interactions. That is what done with immunoelectrophoresis. Then in food industry, it is used for diagnosis of various diseases of kidney, liver, and cardiovascular system. So these are all different applications. Then it is applicable for separation of organic acids, alkaloids, carbohydrates, amino acids, alcohols, and phenols. And in combination electrophoresis in combination with autoradiography. Autoradiography is used to study the binding of ion to serum proteins. Binding of ion to serum proteins. So this all will be very helpful for diagnosing the separation of clinical samples. Then used for analysis of terpenoids, steroids, and antibiotics. Terpenoids, steroids, and antibiotics for testing purity of thyroid hormones. Testing purity of thyroid hormones by zone electrophoresis. Thyroid hormones purity is tested with zone electrophoresis. Then paper chromatographic electrophoresis is used to separate free insulin from plasma protein. Free insulin from plasma proteins. Then it is also used for separation of scopolamine and ephedrine using buffer at pH 4.2. Then electrophoresis is also used for separation of carbohydrates and vitamins. That's what we have seen here also. Then quantitative separation of all fractions of cellular entities, antibiotics, RBC, enzymes, etc. is possible with electrophoresis. These are various applications of electrophoresis. You need to make only the pharmaceutical applications, pharmaceutical and clinical applications of electrophoresis. Okay. So with this electrophoresis topic is completed, mostly all the concepts we have discussed behind that. In the next class, we'll be moving on to introduction on spectroscopy. We'll be moving on to the second broad category of pharmaceutical analysis, that is spectroscopic analysis. Okay, with this electrochromatography, we are completing all chromatographic techniques. Okay. So in electrophoresis, we have seen three kinds of subtitles, that is principles of separation, equipment for paper and gel electrophoresis, then applications of electrophoresis. Okay. Thank you all. Is there any doubt? If you have any doubt, you can clarify now on electrophoresis. So from next class, we will be moving on to spectroscopic methods of analysis. So in chromatography, what are all the techniques we have seen? Planar and columnar chromatography and miscellaneous techniques. In planar chromatography, paper, TLC, HPTLC, PTLC, this all we have discussed. And in column chromatography, column absorption chromatography, HPLC, ion exchange, size exclusion, that is gel filtration, affinity chromatography, and then gas chromatography, this all we have discussed. And in miscellaneous technique, electrophoresis has been discussed now. So with this chromatography, all chromatographic topics have been completed. From next class, different kind of spectroscopic methods will be discussing. Okay. Okay, thank you all. Is there any doubt?
Thank you.
You can clarify now.
Thank you, ma'am.
No, thank you. Thank you. Thank you.
Thank you, ma'am.
Okay, be safe.
Thank you, ma'am.
Be at home only. Don't go outside. Be safe.
Okay.
Okay, thank you.
Thank you, ma'am.